we talked about techniques for examining the proteome, right? Then we talked about how do we examine the proteome in light of the fact that higher organisms like ourselves have post-translational modification, right? So we looked at different things that people are interested in, the major classes, phosphorylation, glycosylation, and ubiquitination. There were some other ones, but the phosphorylation and the glycosylation, those are two of the main ones, because most of our proteins are glycosylated in some way, and then what controls on and off processes in the cell is often phosphorylation of proteins. So if you're trying to figure out what pathways are activated in a cell, say when you give a drug, or finding out what's the difference between a cancer state and a normal state, you're wanting to target things like phosphorylation, see which proteins are being phosphorylated, which ones are active, because that can give you insights into the molecular basis of things like the disease. Kind of leads into the class for next semester, right? Mm-hmm. Molecular basis of disease. Okay. So these, really what proteomics is learning tools and ways to examine all the proteins that are present and ways of actually kind of pulling out clusters of proteins that are actually involved in a disease, for instance. You can actually start to pull out from all these proteins, you can see differences that then you can put a function to. What is it doing to the cell? Okay? So we looked at that, and now we're going to get into the proteomics of protein-protein interactions. Okay? Something called interactomes. What is interacting with what? What are the networks of proteins that are involved in some sort of process? So just in a quick overview, I'll go over what an interactome is. It's pretty simple. It's just what's interacting with what, really. The networks of proteins that are interacting with each other. We'll look at how do we determine protein interaction networks. We'll look at, again, a lot of this is just going over methodology. How do people decipher within this whole pool of proteins what's interacting with what? Why does it matter? Maybe I'll say something about that. Okay, interactomes. What are interactomes? It's basically the networks of proteins interacting with one another in a cell. Okay? So here I'm showing you an interactome. This is the clathrin-mediated endocytosis interactome. So endocytosis, I think we even talked about this with clathrin. That mediates basically the release of cargoes for things like proteins that are synthesized inside, right? They go through the endoplasmic reticulum and then they get partitioned out to different places in the cell. And many of these are mediated with clathrin, right? These proteins that self-assemble around the membrane, pinch off vesicles basically, and will get delivery. So here what I'm showing you is somebody went in and deciphered what proteins does clathrin interact with. So all these lines are being drawn between clathrin, okay, and another protein mediating endocytosis, this AP2. And it's showing that they have interactions with several proteins. The ones like clathrin and AP2 that have lots and lots of connections, we call those hubs. Those are central areas that are connecting with a lot of other proteins. Yeah?
So I noticed in this diagram that there's no protein that all has one connection.
Yes, it's a network. Most of our proteins have connections, at least roundabout, with other proteins. So you can see, for instance, clathrin has a connection here. Let's see here. It's hard to see. DAB here, DAB2, which is also, DAB2 interacts with AP2, okay? And we can see clathrin also interacts with AP2. So this is just showing the web or the network of proteins that are involved in clathrin-mediated endocytosis. We can see things like accessory proteins that have interactions only maybe with a few of these different proteins, but that's what it's trying to do. It's trying to say who's interacting with who so we can understand the networks of proteins that are responsible for a process. Okay? So it's basically deciphering these protein-protein interactions and networks of proteins that are involved in interactions. We'll look at another one later on. Okay, so studying interaction partners. There are two main ways we can study these kind of in vivo or in vitro. So in a test tube versus internally, we can actually start to test these things. And we'll look at different ways out. In vivo, there's actually a few ways basically getting a cell response. In vitro means you purify proteins, and establish interacting partners. So one, we're leaving things in the cell intact and then trying to get some sort of readout of what's happening. The other one is to purify them out and see what's coming along with what, who's interacting with who. Okay? And what we're going to look at are different methodologies for how they study this. The first is an in vivo assay. So there are actually only a few in vivo ones that are really prevalent. There's this yeast 2 hybrid assay. Okay? Sorry. Now, what is the yeast 2 hybrid assay? So in yeast, transcription requires a DNA binding domain that comes into contact with an activating domain. So remember our transcription, these are kind of like transcription factors. This is a DNA binding, DNA binding domain from yeast. Okay? And this here is an activator domain for transcription. So remember, when we're transcribing something, we have to have usually a protein that comes in and binds to the promoter region or slightly upstream. And to this, we usually have another protein that interacts with an activator domain, which activates the RNA polymerase. Now, so what we have to do is, in order to get transcription, you have to have a DNA binding domain in close association or in close proximity to the activator domain. And so what people do or found out is you can take and fuse a protein of interest to DVD, the DNA binding domain. So this is a protein fusion, covalently linked. So we took a gene for this DNA binding domain, and we fused a gene for this target protein that we want to understand, does it interact? What does it interact with? Okay? So we're question. This is our question. Who does X interact with? Okay? And what they do is they say, okay, if we have a DNA binding domain and we fuse a protein to it, a protein that will interact with it, if we fuse AD to it and it does interact with it, what will happen? We'll have a reporter gene downstream that will get produced. Okay? And they usually use beta-galactosidase is the usual reporter gene. So if you fuse this protein that you suspect, okay, we suspect it interacts with X. This is our suspect protein. If we fuse it genetically to the gene for the activator domain, these will be expressed in the cell. The DNA binding domain will bind here, If it doesn't interact with it, we don't get any transcription and translation of a gene product. However, if they are interacting partners, what will happen? Their interaction will bring the DNA binding domain and the activator domain into close proximity, allowing RNA polymerase to make a transcript, and then we'll get a protein that is expressed. The protein that gets expressed, so we get transcription, that gives us a protein, usually an enzyme, and then so we can visualize through a colorimetric assay. Colorimetric. And I'll show you an example. So does this make sense? They've discovered in yeast that in order to have transcription, you need to have a DNA binding domain that comes into close proximity of an activator domain. We can actually split the genes for these apart and put other protein genes on them. One that we are basically kind of baiting this protein with, and this is our prey. If the prey comes into the bait, we activate transcription. If the bait here doesn't bring that in, that prey, well, we don't get any transcription, so we would say no interaction between those two proteins. If we do see the response, we would say, oh, they form an interactome, an interaction with each other. Does this make sense?
Now, couldn't this have a problem of when you bind it to the DNA binding domain, it could cause changing of the...
Yeah, that's something you have to worry about, so you have to make sure you do controls that will check for that. But for the most part, they found that this is a pretty robust system. As long as you have a good linker here, the DNA binding domain is fine. It won't change the structure of that. Now, it could screw up the other protein, but we can't know a priori beforehand whether it's going to do it or not. But for the most part, they found that this can work. And I'll show you some examples. So here is a yeast 2 hybrid in practice. So the gene down here is usually beta-galactosidase. So our reporter gene, beta-galactosidase, is the typical one. And what it will do is it will take a substrate called X-gal, and it will convert it into a blue dye.
We mess with that in animal.
Yeah. And so here's an example. We have different, partners that we're baiting. So we have a protein that we are baiting some prey, and we have different preys here attached to an activator domain. The gene that will get activated downstream is beta-galactosidase, which does this reaction that takes a soluble, colorless substrate, breaks it apart, and makes an insoluble blue dye. Okay? So here we would say, this is kind of our control, or no, this is one that we're not even. So here would be our control. This is basically the protein without any prey. Okay? So this is what you would normally see. Okay? This is a negative control. We're not seeing any blue, so the protein is not being produced downstream. Okay? So this is our negative control. Now, this is just looking at different fragments of proteins that they think interact with this GBK protein, GBKT7. Okay? If it does form an interaction, what happens? Well, you get transcription of beta-galactosidase, and you see the blue colonies, yes?
So section D is the full-length DNA strand?
No, full-length protein. They're trying to decipher where the interaction occurs.
But even then, that says from 1 to 471 amino acids?
Yeah.
But then why is there 1 to 504 in ENF?
This is a...
I mean, that's
these are mutant. This is HDA9, which is different than 6. So these are different So these are two different proteins.
Which is really simple.
Yeah, so this HDA6, they're looking at full length, and then they're looking at fragments. Then they go to this HDA6-9, which is a different one, a mutant one, and they want to know if it interacts with that.
Actually, what's the difference between E and F?
What? ENF.
Yeah.
It doesn't say. Doesn't matter. You get the idea. It's just for a conceptual thing. Okay. So you can see for this, we can see the full length interacts with its bait. Okay. And then we can see even amino acids 1 through 131 interact, right? And then this last fragment here, this 1 to 467, this is probably a mutant that they made. It destroyed the interaction. Oh, this is 10, HDA6. Yeah, they've truncated this one slightly.
Wow, I'm at like 4.
Yeah, must have messed up its structure.
Weird.
But do you get the idea? Not the it's not about the specific type. It's that, oh, if you look at these, if it interacts, you can see this readout.
So then you're narrowing down what section of the protein is interacting?
Yes, you could do that, but that wouldn't really work all the time because if you truncate it, you throw off the whole structure.
Right.
So they're probably making mutant forms where they want to see, oh, C-terminal end, does that have any influence? You could imagine cutting off a C-terminal end. Yeah, that wouldn't make much difference. So I would say, based off of these two results, that the C-terminal is essential because you lose it if you just truncate.
Would this be like seeing where they could modify the protein to produce a different effect, where they could modify it for a drug use or something like that?
I would say there are probably ways you could do that, depending on how you design it. You could put one with drug and one with no drug, and if it doesn't allow them to interact because the drug causes a blocking of an interaction, you would see only activation of color when the drug isn't present because it blocks the interaction. Whereas if you grew them up on medium with drug and that drug caused a conformational change that doesn't allow the bait and the prey to interact, you wouldn't see it. So you could probe things like does drug cause a change in interaction between two proteins? Does that make sense? Yeah. Okay, so this is one of our in vivo methodologies. There are downsides, like I say, attaching another protein to another protein doesn't always lead to proper folding and things of that nature. So, but it works, and it's only in yeast, right? There are things that we would like to look at that are outside of yeast. Another one that's used, this is more, they call it an in vivo in your book, but we want to know which protein interacts with protein X, let's say. So you can take protein X out of the cell, okay? You can put it on a surface, okay? We want to know what interacts with this protein. Well, you can take phage, okay? And phage, you can modify their genome. You can insert genes into their genome, and this will display, okay, the protein that or peptide that you put into their genome. It will display it on the outside of the phage, okay? So you can think about putting proteins on the outside of the phage. That doesn't always work. But the deal is you can put lots and lots of different protein genes in these phages, and you can get a whole library of phages with lots of peptides and protein fragments displayed on the surface. Then you can have your phage, okay, mix it with your surface. If the phage sticks, okay, you can wash away the other ones. You can take that phage and have it infect cells, and the phage that get infected, we can actually sequence the genome from those phages that make it through, that get propagated in cells, and we can figure out what protein or peptide was on that phage that caused it to bind. Does that make sense? So it takes you basically recover the bound phage. Usually you infect cells so you get more of that phage built up, and then you sequence the genes that are in the phage. And then you can figure out what was actually interacting and binding. The problem with this is usually it's not large proteins. It's usually peptide fragments. So you might get peptide fragments that you could identify to a protein. But other than that, it's usually not large proteins. But it is This is what they recently got the Nobel Prize for, was developing, in chemistry, developing phage display. Yep.
Are these peptide sequences only like 10 or 15?
Yeah, typically they're on the order of 8 to say 12. You can do bigger ones. The problem is the longer the sequence, the more potential genes you have to make a library for. The longer the genes have to be. So, but you can do libraries of proteins, but the larger the peptides get, the more different phages. But phages are small, and you can have millions of different phages. All you need to do is pull out the one that binds. Okay, so that's phage display. So you guys understand the two hybrid methodology, and then we have the phage display. We're going to look at some more that are in vitro. Okay, so another one is co-immunoprecipitation. Basically, you can take beads or a surface and modify it with an antibody that's specific for a protein you're targeting. So this antibody is specific to a protein target. Specific to a protein target. We'll call that the bait. Okay? And the prey is what protein is actually going to interact with it. We're trying to figure out what protein interacts with it. So you have an antibody that's specific to your protein target. You can immobilize it on any surface, but we're saying beads here. And here is our bait protein, the red, that the antibody actually binds to. Okay? Now, if it binds to the antibody and there's a prey protein that also interacts with it, or there might be other prey proteins, what will happen? They'll be immobilized with the bait. Yes?
I'm probably thinking about this way too hard, but wouldn't we call it a predator? Oh!
Yeah, it's baiting.
We are preying on the protein.
Yeah,
okay.
we're trying to figure it out.
Okay.
Yeah.
Sorry.
So here's our bait to bring in the protein that actually interacts with it. If it gets immobilized, the protein that has a protein-protein interaction with it will be immobilized as well. We'll wash away any proteins that are not interacting with it. And then at the end, what we can do is elute this off. We can run a gel, for instance, elute it off, run a gel. We can see that there are multiple bands, and we can use the methodology that we've talked about to identify what was the prey protein, what was interacting with it. Yeah.
Is the little green thing also a bait? Or
It looks like it's a bait
prey?
as well.
Okay.
Yep. So that one's a bait as well. Or it's a, that'd be the prey. That would be a prey protein. as
So
well.
like two preys
Yeah.
on the same bait?
Yeah, because you could have multi-protein complexes, right? Not just one protein with another protein. You might pull out 10 proteins with one protein that enters. Yeah.
What's the bait protein?
What?
What's the bait protein?
The bait protein is the protein you've identified as interesting to you. And what you're doing is saying, okay, what does it interact with? We've identified this protein. I want to know who is it interacting with. Maybe it's a protein tied with regulation of a cancer that you've found out. You want to know, okay, this is regulating cancer so that it's out of control. What is it interacting with downstream? And then you can basically pull it out, basically elute off wash away all the proteins that don't bind, elute them off, separate them out in the digest, and then you can figure out what their identity is using the methodology that we've already talked about in mass spec.
So are we targeting the bait or the prey protein?
What's that?
Are we targeting the bait or the prey protein? We cannot target-
We're targeting the prey. We're baiting the prey.
Yeah.
We know what the bait is, and we want to know what is it interacting with. So the prey is going to come in and interact with the bait, right? So we know what the bait is, but we're like, what's attracted to the bait? That's what we're doing. Where is the protein-protein interaction? There are proteins that are attracted to it that interact with it. Who are they? So we can protein ID, and then we can validate this with Western blot. Does that make sense? Okay. Here's another one. So immunoprecipitation requires that you have an antibody for your protein the bait of interest, okay? And it precipitates things out, okay? But there's another method that's fairly similar, but you don't need necessarily an antibody for this. You can do what's called a protein pull down. You need some tag on your protein of interest that will fish it out of solution. So here's an example. You can have these different proteins here, and they can have even different methylation, what have you, that we've talked about before. And you can modify them with an affinity tag. You can do this synthetically, like we've talked about. You can put an avidin tag on it. We talked about methodology for targeting things like sulfurs, and we can put a tag on that. And avidin will bind to biotin, right? So the tag is actually the biotin. It will bind to avidin, okay? And we talked about the columns, right? The biotin, streptavidin column interaction. So you can tag these proteins, okay? And then you want to know, well, what interacts with this protein that we've tagged, okay? Well, you have these. You can take nuclear extract. If the protein binds to it, you can immobilize it on the avidin column because this biotin causes it to stick. And then you can see, under different conditions, what have you, you can pull out your different complexes and you can say, oh, here's our protein. Oh, we pulled out another protein under this even lysine methylated condition. Here's one where the lysine 9 is methylated. So we can look at even different post-translational modifications and see what interacts with this target protein when the target protein even has different post-translational modifications. So in this case, we didn't see anything interacting with it when it wasn't methylated. Well, we looked at a condition that produced methylation at this lysine 4, and then we saw we pulled out this protein here, this BPTF. Oh, we can now cut that out of a gel and see what was interacting with it. Use our bioinformatics, work backwards and figure that out. Or if we methylate K9 here, right, we can pull out this HP1, pull down, right, because of this tag on here, it will stick to avidin to that bead, and it will be immobilized. Everything else gets washed off that doesn't stick, and then when we run our gel, we'll see a new band that emerges here, and we can identify that.
Is avidin sort of like the membrane that biotin binds to?
No, it's a protein that it binds to. Avidin is, the biotin is the small molecule, avidin is a protein, and they have one of the tightest affinities of any complex in biochemistry. It's like 1 times 10 to the negative, or 1 times 10 to the negative 16th molarity KD. So it's like super, super tight. So that's why they use it. You tag your protein here with biotin, and then you put it on a column or with beads that have avidin on them. They basically stick very tightly. You can really wash away any proteins that don't stick. Now, you do have to be careful because some proteins only interact transiently, right? This is assuming that when you're doing your washing, that the protein stays. But protein-protein interactions are concentration dependent, right? So if you dilute it too much, you might never see the protein. Okay, so there are other techniques we'll talk about here, such as chemical cross-linking. So is everybody okay with the pull down? You can do things like His tags. You learned how to purify proteins with nickel affinity chromatography. So you could His tag a baked protein, pull it out of solution with the nickel affinity chromatography, and then see what's attached to it. That's another way of doing it. So you can, engineer a tagged protein. All right. Another thing that you can do, there's that problem with transient interactions, okay? Some proteins interact, but it's concentration specific. If you start diluting them with these processes, what happens? They dissociate. So to get around this, what people will do is they'll cross-link the proteins. They'll add molecules that will physically, covalently cross-link any proteins that are in close association with one another. Glutaraldehyde is a very common one. We'll look at some other ones. But basically what happens is you cross-link them first, Then you purify them out like normal on a gel or whatever. And then you perform an enzyme digestion. You get fragments where there are now peptides that are cross-linked with one another from different proteins. Okay? Because they were forming a non-covalent interaction, they got cross-linked. From these, we can get peptide masses, and then we can use our bioinformatics, and we can now determine interactomes. What peptide fragments came from what protein? Who are they interacting? This is just some examples of cross-linking reagents. There are specific cross-linking cross-linkers, we call them. Things like succinamide esters. These cross-link two lysines that would be in close proximity to one another. So amine-amine interactions, and this is what they would do. If you have two amines close together on a protein in space, this will come in that can attack these different parts, and you permanently cross-link them. Okay? You can have carboxyl specific, so if we have two carboxylates next to one another, okay? You can actually cross-link them with things like these hydrozides, hydroazides. See, this would attack here, this would attack here, and then we can cross-link them. There are also other things that will cross-link amines to carboxylic acids, which makes more sense, right? You have two proteins in close proximity to one another, okay? You would probably have a carboxylic acid next to a lysine on the surface of a protein. I'll go into a little more detail here in just a second. But there are things like EDC. We call it cross-coupling. EDC will do this. It will take this amine and it will basically cross-link it to a carboxylic acid. Now, this means that these functional groups would have to be pretty close right together to cross-link them. But what you can do is you can use cross-linkers that have different numbers of spacers between the reactive parts. So, well, why would lysine be next to lysine? Well, they might be close in space, and if we use a long enough cross-linker, they'll be close enough that they can cross-link. So these have spacers that we can modify their length with. Okay? And what we would say is the shorter the spacer, if we get a protein that cross-links with a short spacer, we would know for certain that those proteins were really associated with one another because they had to be close enough to do that cross-linking. Over here, so these are adjustable cross-linkers, so you can change the space, the distance, to give you more flexibility in where you label. There are other ones called zero length. In this case, we use zero length because these should be close together in space, so we want to use a cross-linker that cross-links, that doesn't take things that are far apart but is very close. So they call them zero length. There's no spacer between them. Does this make sense? Yeah. So we can add cross-linker to the cell or whatever to our extract. It will permanently lock them together, and then we can digest them and use all the things that we talked about before. Those are cross-linking reagents. Okay, another method. These are all in vitro, okay, that I've been talking about. We have to break open the cell and then test it outside the cell. Yeast was really the only one that was in vivo. Another one is FRET, fluorescence resonance energy transfer. You'll hear this being called FRET. Now, how FRET works is if you have proteins that you label with a molecule that will absorb light, okay, and then it can do fluorescence. If you label another molecule that can accept the wavelength of light that fluoresces, you can see whether they're close enough in space with one another to be forming an interaction. Okay? So if you have two proteins, okay, that you've labeled and you want to know, are they forming a non-covalent interaction with one another, we basically label one with a donor molecule. It will take light, absorb it, and then it will fluoresce. The molecule here is one that accepts that So that's an acceptor, that wavelength of light that it absorbs, and we can see whether that quenches the fluorescence that comes off of there. If they're not close enough, right, we know they're not interacting. So if they are close enough, they're forming a non-covalent interaction. The fluorescence from this will get transferred to that fluorophore, that acceptor here, and we would see that it would show up in the spectrum, the emission spectrum. So it would absorb that. So you see here, we're looking at 620. These are not close with one another, so it's emitting this light at 620 that this accepts, but we don't see anything from that because they're not close enough in space. Has to be something like less than 10 nanometers to have fret. I think it's less than 10 nanometers. So you have to be within 10 nanometers for fret. If they are close enough, we get fluorescence resonance energy transfer. This absorbed light, and instead of dissipating it off as fluorescence that we would see emitted off, it goes and gets absorbed by another fluorophore, and we see the emission of that fluorophore show up because they're close enough. If they're not interacting, they can't have that transfer. Have you all heard of fret before? No. But what's that?
I've heard of it, but never actually.
So this is a fret. Again, you have to label this would be kind of your bait and your prey. Does it interact? You have to label it.
I mean, I fretted over your test.
Yeah, that's good. That's what I hope for. I hope you all worry and fret. All right. So those, you have to prepare. Those are a little more difficult, but doable. Okay. Now, what about these transient kind of interactions? You've had something. We talked about cross-linking them. And then if they have a transient interaction, well, they'll be permanently cross-linked, and we'll see them by mass spec. There's another way of doing this. There's what's called label transfer. We have protein 1, okay? We can actually label it, okay, with a transfer label. So it has something, an identifier attached to it. So we can basically cross-link the protein 1 with part of this, we'll call it a label. Okay? So we get a protein now that's labeled. And part of this label has a disulfide bridge, which we can break by putting in a reductant, right? Something like DTT, beta-mercaptoethanol, breaks disulfide bonds. So we've labeled a protein, and we want to know, does it interact with another protein? Well, we have a reactive group here, an azide, that will react with proteins that come into close proximity. So here comes protein 2, okay? And these are UV activated. So this is UV activated. This will react when we activate this by shining light on it. This becomes reactive. If anything's close by it, attaches to it covalently. So we do this process. If this protein interacted, it gets cross-linked. Now we can reduce this disulfide, and what now has the label? The protein that it interacted with. And so we can decipher that out. So basically, what you're doing here is you tag a protein with a label that is reactive and chemically transferable. Okay? You cause interaction leads to labeling of another protein, and then you reduce the disulfide to send the label, with the protein interact. Does that make sense? So this, we're getting kind kind of creative with how we're chemists, right? So we can devise tags that we can cross-link to a protein, and that if it cross-links with another protein, we can cleave it off and send it with that protein and identify what it is. Yeah.
So I'm guessing protein 2 wouldn't naturally interact with the tags?
No, not naturally. This gets activated by the light, so it makes this very reactive, and then because it's in close proximity, it will react with amino acid residues on the protein or even the backbone.
And you need the tag attached to protein 1 to make that happen?
Yes, because then it wouldn't come into close enough proximity
Oh.
to it to react. So does that make sense? See how we can get kind of creative with how we see what has interacted with what? Okay. I think this is the last one. Yeah, this is the last kind of experimental one. This is tandem affinity purification. And how this works is you're doing a tandem affinity purification. There are two affinity chromatographies that are done in this process. So that's why it's called tandem. Usually what you have here is a bait protein that's fused. This would be genetically made, okay, with a calmodulin protein. Calmodulin or calmodulin tag binds to calcium, okay? Then you have a spacer, okay, that has another protein, protein A, which we have an antibody for, okay? So we have this cell extract, and we have this protein that has a bait protein on it. Okay, we can mix it with our cell extract, and then what we do is we have a first column that has beads that are covered with an antibody that are specific to protein A. So it immobilizes that. Okay? Then we can wash away anything that's not interacting with it. Okay? Then what we do is we can clip the TEV with a protease, the tag, the A tag, so that now we have a soluble protein that's not permanently bound, and then we bind it to a second affinity chromatography column that binds to calmodulin. So it has basically calcium on it. Calmodulin beads. Okay? And then basically here, we've now immobilized this. We can use a kind of, I don't want to say less harsh, we can use a washing protocol that is mild, a more mild washing protocol, and then what elutes off is the protein with whatever it's associating with. And we can identify with mass spec, all those things, right, the proteins that are associated. So they're using EGTA to elute it. EGTA binds to calcium ions. So if you take away the calcium ions off the column, it won't. The calmodulin won't bind. All right. Does that make sense? Okay. So those are all kind of the experimental techniques to start to decipher these in silico or in these interactomes with one another. This is actually the most high throughput of the ones that we talked about. This is actually very high throughput. You just can pool, take cell extracts, throw it with this, and you can run these processes fairly fast. The other ones are more time consuming. Although cross-linking is not quite so bad, I should say. That actually is a pretty high throughput as well. Computation. So we not only can decipher interactomes experimentally, with wet lab chemistry, but you can also use computation. And there's this process called Rosetta Stone protein analysis. And how this works is, let's say you have two proteins in a genome. Protein A, protein B. Okay? And they're separate in the genome, and then we have a question. Does A interact with B? Okay? So what you can do is you can look at these gene sequences, put them together, and see. So let's say this is organism A. You can say, okay, do I find anywhere in nature, in our genome, do I have, or can I find a gene fusion of these two domains? Okay? So what I'm doing is I'm saying, okay, there's these two proteins, and I want to know, do they interact with one another? Well, if I look throughout organisms, I might find a protein that is really the fusion of those two genes. And that, you know, the assumption is that maybe these got separated at some point, okay? But originally they started out as one gene because of like the evolutionary theory, right? Yeah.
Is it possible for the DNA just like loop back and then them interact that way?
No, so what it's saying is these are two separate genes in an organism. And the question is, okay, do these interact? Well, if I could find a protein in any other organism that basically has these gene products fused together to make another protein, they probably interact. So you can basically, in silico, you can fuse these two genes together, and then you can screen genomes, and you might find a genome, organism B, that has this. So we identify. And then what you would say is, from this, A and B likely interact. Sorry. Does that make sense? So we're just looking at a genome, and we're like, okay, we have organism A, and we don't want to do all these experiments. We have the genome present, and I've identified two genes, A and B. And the question is, do they interact with one another? Well, in silico, means on a computer, we can splice these two things together and see if we screen genomes, do we find anything that matches? Genomic screen. If we find a match, so we can just take those two and we can make a theoretical gene fusion of the two. Then we can say, do we find a match? Okay. If we do, we would say, oh, A and B likely interact with one another. If you see two proteins in a genome where those two domains or those two proteins are fused together, you can say, oh, those likely interact. Because, you know, through the evolutionary theory, they would say, oh, at one time maybe they started together and then they broke apart. Does that make sense? So it's just a way to screen through and see if you can find them. So that's kind of how you could do it, just computationally. And there are other ways too. There are things like docking, which is more high-end. So there are others. There are things like docking. So if you know this structure, you can say, oh, screen to see if there are any proteins with known structures that could potentially dock into this protein. It's a little more high-end, but that's another thing that you can do. But this one's pretty straightforward. Okay. So interactomes. What are we trying to do with interactomes? We're trying to decipher molecular pathways coordinating biological functions. So you might want to know what proteins are related to, I don't know, colon cancer. You could find proteins that interact to cause that disease. Or what if you want to understand how is the sex of an individual determined biochemically? Well, there has to be interactomes, right, of proteins involved with that. Or not even in humans. This could be in things like fruit flies. And I think there's something like this in your book. But you might want to know fruit fly sex determination. Well, you could evaluate interactome of proteins responsible for this. And now you get these clusters, just like the clathrin that we talked about at the beginning. Interactomes help to inform us of what these proteins carry out biochemically, what processes. Does that make sense? So interactomes are really clusters of proteins that carry out specific processes. Now those can be linked to other processes, but that's how we can think about them. Here's an example. And this is just showing how even these interactomes are conserved. So here, this would be like an interactome, multicellular development, right here. Okay, these proteins are involved with multicellular development. Okay, that would be an interactome of a process. Okay, here's some for growth regulation. Now, what's interesting is different life forms actually have conserved interactomes. Okay, we're looking at two of them here. This is endocytosis, I guess, because everybody's interested in endocytosis. So the blue one here, if I remember, is yeast, and the other one is for a worm. So genes. So this is our yeast side in blue.
It says yeast is in green.
Oh, yeast is in green.
And worm
Yep.
is blue.
Yeah, okay, that way. So worm is blue, yeast is green. And what we see here is that between them, there are conserved interactomes in red. So we can even identify processes that between organisms are very, very similar. And how would this be useful? Well, we can't, for instance, in humans, can't do a lot of medical testing on people, right? We can't do a lot of research on individuals because there are ethical reasons for that, right? But if we find interactomes that are similar with these lower organisms, right? Well, we could study things through their interactomes that are similar to us. Does that make sense? So this is why this is important. If we understand these networks and similarities between different organisms, where there are things that are similar, you could use model systems and make connections between us and those for things like treatments or progressions of diseases, things of that nature. So some of these processes are highly conserved, but you can see the differences between them are important. That's what differentiates organisms, really. And this is something I think your book says is the complexity of an organism isn't the size of the genome. It's not the size of the genome. We saw that the bean plant has way more genes than we do. It's the number of interactomes. The more interactomes you have, the more complex an organism is. So complexity, okay, is related to the number of interactomes. What's that?
Oh, yeah.
Yeah, it is. Looks like happy. Does that make sense? Okay. And that's all I got. So hopefully we understand now what interactomes are, the significance of them, if we want to decipher pathways that are important for cell regulation, cell death, all these kinds of things. We want to know what interactomes there are. If you look at some of the cancers, there are these enzyme cascades, or there are protease cascades, these Cas spaces. And these are often involved in apoptosis. Well, what happens with a cancer? Apoptosis kind of goes out the window, right? It's not being activated. So you want to understand, okay, what is happening to the apoptosis machinery, right? And if we understand the interactome there, maybe